Here we are again. We are ready for our next podcast and last one with our biochemistry unit. And as you can see, we're going to take this idea of these biochemicals, these organic compounds that make up our bodies and that we consume all the time. We're going to take that and apply it to what happens when we consume them and what our bodies do with them. So we're going to talk about the biochemistry of digestion today. First thing we'll focus on is why it is that we need food, and we've touched on this before, but let's remember that all of life needs food, organic compounds, to physically build their bodies. My body is made of organic compounds, and so if I'm going to build a house, I need the bricks to do it. Well, if I'm going to build a body, I need the organic compounds to do it. So it's raw materials, those amino acids, sugars, nucleotides, particularly amino acids and nucleotides, will go into building my own body structures. Additionally, we need food as an energy source. Our bodies, just because we have a pile of bricks, doesn't mean they're going to spontaneously jump up and build a house. We have to put energy into that. And so just like that, in order to build our bodies, we have to have energy to do that, and then of course energy to do all the things we want to do once our bodies are built. So we need organic compounds along with some oxygen to make an energy source that our bodies can use, and that energy source for us is called ATP. We'll talk much more about that later. Don't worry about it now, but just understand that we need energy to build our big macromolecules in our body from these raw materials that we take in. Okay? Now some organisms have it made, so to speak, in that they make their own food. This is an autotroph. It is a photoautotroph, this tree doing photosynthesis and making its own food. However, some organisms have to eat their food, take it into their bodies from the outside. They can't make it within their own tissues. So the snake is having a meal. Okay? Right now we're going to talk about the ones that have to eat food in this in this podcast, like us. And I mean especially us, because this podcast is going to focus on human digestion and what we do with those biomolecules, those macromolecules that we take in. Okay? Specifically, we want to talk about not just what we take in, but what happens to it, what acts on those molecules once they are taken in, and what we end up with at the end of these chemical processes. And we want to talk about where all these molecules will be found in various places in our digestive tract. So, there are two main processes in human digestion. They are mechanical digestion. That's not a human, but he does pretty much just like this with regard to this. He's doing mechanical digestion. He's physically breaking apart food molecules with his teeth. We do it with our teeth, with the churning of our gut. So mechanical digestion is very simple. Chemical digestion is where we actually use chemicals to help us break down food into even finer molecules, smaller molecules than what can be achieved with mechanical digestion. And this gets a little bit more complex, and that's what we're going to focus on for the rest of this podcast. So, what things do our bodies need to chemically digest? Okay? We need to digest organic molecules, like maybe this cracker, or like maybe this cheese, which might have some protein and fat, and this cracker might happen to have some carbohydrate in it. Okay? So we got to break down things like starches, carbohydrates. It's not that we don't take in sugars. We do, of course we do. But those don't need as much breaking down because they're already reasonably small. Starches need a lot of breaking down because if you'll remember, they're huge molecules made of many hundreds or thousands of glucose molecules. Lipids also need to be broken down before our body can deal with them properly. Proteins also need to be broken down before our body can deal with them properly, as do nucleic acids need to be broken down into their monomers before our body can deal with them. So all of these things need to be broken down into smaller and smaller molecular forms for us. Okay, now let's do a little anatomy really quick. This green shade to this diagram is a little disturbing, but at any rate, I want you, this digestive tract looks really complex. If you look, it's almost hard to tell what happens. We can kind of follow this. Food would go in here and go down this esophagus, and then we can go into the stomach, and when it leaves the stomach, it's like, what happens? What you need to think of the human digestive tract. Basically, you need to think of it as a hollow tube, just like that paper towel tube, just like this tube, that if I looked up, if I looked through it, I could see all the way through. I could see you. Okay? It is a tube. It's just that the tube of your human digestive tract is folded up and coiled a lot, so we can. It's a recurring theme in biology that we often have to fit big things into a small space. This digestive tract is really long. It is huge. It is also referred to, by the way, as an alimentary canal. Same thing. Digestive tract, GI tract, gastrointestinal tract, alimentary canal, all the same. Do not let that ever confuse you. But it is extremely long if you stretched it all out. You can see we're starting with the mouth here, ending up at the anus, and it is a long tube in between. Okay, 100 centimeters. Right there is a meter. And so if you'll see, this thing is doubling around many times, so it's several meters long. So what's happening is, let's orient ourselves. As I said, this is the esophagus we're looking at right here. In the stomach, that's where the food ends up first. So mouth to esophagus to stomach. And you can see that makes up a pretty small part of the digestive tract. We are going to exit the stomach right there and go into the small intestine. Now, this first part of the small intestine here has a special name. It is called the duodenum. It's small intestine. It's just that this first little part has a special name. Okay? And in this diagram, the duodenum is starting right about there, and then it's kind of hard to see because it's hidden a bit by the large intestine. Now, the duodenum is right next door to the pancreas, which is not part of the tube. It's not part of the tube. It sits outside the tube, like my fist is sitting outside this tube right now. Okay? But it does squirt things into the tube. It's got a little duct that enters into the tube and will squirt digestive enzymes in there. Now, once the food passes we take in some food, goes through the stomach, goes through the duodenum, it is now in what we call the jejunum of the small intestine, and that by far is the longest part, and I would tend to disagree with this diagram. I don't think I think this jejunum goes quite a long way, but whatever. The ileum is the last third of the small intestine. So jejunum, I would say, is probably most of this stuff. Ileum, probably down in here. All of that is small intestine, which is where most of the absorbing of the actual nutrients come. We're concerned mostly with breaking nutrients apart up in this part and absorbing them into the bloodstream through this part. And then after this we have the large intestine, which we won't talk too much today about, but that's where basically feces is stored and so on until we can get rid of it. So but it's a hollow tube, and that's the bottom line of what you need to remember. Okay? All right. Now, most chemical digestion, not all of it, but most of it, is going to be occurring in the mouth, a little bit, in the stomach, a little bit, vast majority of it in that first part of the small intestine that we call the duodenum. This little drawing has had the stomach taken out and the liver taken out so that you can see things a little more clearly, but obviously you're missing stuff. So the stomach would be going up here. I'm no artiste, but this would be the stomach, esophagus sending food here, and then into the duodenum, through the stomach into the duodenum, and then the liver would be kind of up here. Okay, now let's go through what happens in each of these organs with regard to chemical digestion. First thing, what gets digested in the mouth is mostly just carbohydrate. Lipid, protein will get mechanically broken down and moistened by saliva, but they will not get digested to any great length, at least not chemically. And when we say what gets digested in its carbs, it's mostly polysaccharides. We're not going to break down those teeny weeny little disaccharides yet. The only enzyme you have in your mouth for breaking things down is going to be attacking the big polysaccharides like starch, maybe a little bit of glycogen, but mostly starch. Okay, so what does the chemical digestion in your mouth? It is an enzyme indicated by the blue here called amylase. Amylase will hitch itself around those glycosidic linkages in starch and break the big, huge polysaccharide of starch into smaller hunks. It's still polysaccharides, it's still big hunks, but it's smaller than it was. Okay, so the products that result are simply smaller polysaccharides. Now, that would happen in your mouth. Swallow that along with whatever, if you had cheese and cracker. Okay, yum. I'm going to chew in your ear. How annoying is that? The cracker is already being digested in my mouth by amylase, but the cheese is not. It's protein, that's lipid. That's going to happen down in the stomach. So in the stomach, mostly it's just protein that's getting digested. There's no enzymes created here to break down carbs. There's no enzymes created here to break down lipids. Proteins, however, can be broken down in the stomach. What is doing this breakdown is our enzymes collectively called proteases. Remember, ACE means it's an enzyme. Protein is what it's breaking down. Now, the specific enzyme that is at work in the stomach is this globby looking thing here called pepsin. Okay, and the products that result when pepsin acts on the proteins in my cheese is just a bunch of smaller polypeptides. Again, very much like with starch, we're not going to break it down into the little bitty amino acids yet. We're just going to break this huge protein into smaller chunks that are still kind of big, but they're smaller than a fully functional protein. So, small polypeptides. Now, we need to talk a little bit more about pepsin, because there's something that's true about pepsin that is true about several different proteins in living things. Pepsin is an enzyme that belongs to a group of enzymes called zymogens. Okay? And zymogens are simply enzymes that have two forms. They have an inactive form and an active form. Pepsin is one of these. Its inactive form that is actually made when inside the stomach, the cells of the stomach, is called pepsinogen. Okay, and that is inactive. And it only turns active and becomes the active form we know as pepsin when it encounters the hydrochloric acid, HCl, that is also in the stomach. And when it hits that acid in the stomach, it pepsinogen converts into pepsin and then begins breaking down proteins. Now, why does it make sense that pepsin is a zymogen? Why would that be a good thing? Well, what is the stomach? It's muscle. What's muscle? It's protein. We don't really want our stomach cells to be creating something that is going to break them down immediately once it contacts them. So initially it's inactive, and then once it is secreted out into the chamber of the stomach, then it hits that acid, becomes active, and we can begin digesting those polypeptides in the stomach, those proteins in the stomach. So, question is, well, fine, it's still contacting stomach tissue, isn't it? Why doesn't the acid and the pepsin just eat holes in the stomach muscle itself? And the reason for that is there are mucus secretions in the stomach that line the stomach and protect it from these things, the acid and the pepsin that could digest it. There can sometimes, however, be problems with the mucus secretion. It gets eroded, and that usually is due to a bacterial infection, and you can have an ulcer as the bacteria wear away the mucus, and then the acid and pepsin start eating away at the muscle itself of the stomach. Hydrochloric acid, by the way, also serves to kind of kill bacteria that you might eat. So that's another use for hydrochloric acid. So it's very beneficial to have it there. Now, the place where the vast majority of chemical digestion occurs is this first part of the small intestine. So here the food comes down from the stomach through the esophagus, down from the mouth through the esophagus into the stomach. It becomes mixed with the hydrochloric acid and churned up and becomes this liquid of digested food that we call chyme, and it's acidic, and then that acid chyme is passed into the duodenum. Okay, there's a whole lot going on here. So it's receiving that partially digested chyme, but the duodenum is also getting lots of different chemicals from the different digestive glands. Mainly, we are talking about the liver and the pancreas. Okay. Now, just so you're clear, what we've got is the stomach dumping food into the duodenum. We also have the liver here, big orange thing, dumping stuff through ducts, and it's going behind the duodenum and through here and dumping chemicals into the duodenum, and also the pancreas dumping chemicals into the duodenum that are going to meet that chyme, that partially broken down food. Okay, this is essentially the same diagram over here. It's just showing you more of the ductwork inside of the pancreas itself. Now, carbs that we broke down with amylase, the salivary amylase in our mouths, are going to get further broken down here because they're going to meet with pancreatic amylases, meaning amylases that are made in the pancreas. Amylases will be dumped in here, and any carbohydrates that are in the duodenum from the stomach will get further broken down into smaller polysaccharides, maybe as small as maltose and other little disaccharide components. Lipids in the duodenum. Fat globules are not getting digested up to this point. Remember, they are nonpolar. Lipids are nonpolar. They're not watery things, are not terribly interested in bonding with them, and so they make it fairly intact down to this point, even through the stomach. It takes special secretions from two organs, guess which two, the liver and the pancreas, to handle these lipids. The liver is going to make bile. So the liver is up here, let's say, and it is making in its tissues and its cells bile. This bile is secreted through a bile duct. It is stored actually in the gallbladder for a time, and then will be released from the gallbladder into the duodenum. Okay, this bile does not break any bonds in fat molecules. What it does do is make the big globs of fat into smaller droplets. We call that emulsifying. It's not breaking bonds. It's just kind of making it into smaller globs to deal with. So emulsification is a word you might see. The pancreas, however, is making enzymes. So remember, the pancreas has already made us some amylases to further break down carbohydrates. It is also making lipases that act to actually break covalent bonds between those fatty acids. Remember the E molecule, the fatty acids, and the glycerol. It's going to break those ester bonds. Covalent bonds between those so that we can further process those lipids and use them. Now, proteins are also handled in the duodenum, and they were handled a little bit in the stomach as well, but we're going to handle them better and further in the duodenum. The small polypeptides that are coming at us from the stomach are going to get further broken down. There are lots of enzymes that do this. Trypsin, which is also a zymogen, and you can see that here. Its inactive form is this trypsinogen, and that's what's made in the pancreas. But again, we don't want to break down the pancreas itself, so it's inactive, and it only becomes active when it enters into the environment of the duodenum. Essentially, what is happening is it's hitting chemicals in the duodenum, the acid, chyme, and so on, that will convert it to its more active form. It's going to break the large polypeptides into smaller ones. There are other enzymes. Chymotrypsinogen gets converted to chymotrypsin. That's essentially doing the same thing. These two are essentially doing the same thing. This procarboxyl peptidase, you're looking at that and going, oh, Coleman, what are you doing to me? But just get that. Peptidase means we're going to break peptide bonds. That's the inactive form. You don't need to know it. This is the active form. And look, there is also an aminopeptidase. Remember, aminocarboxyl on either end of the amino acids. This can break the bonds on the amino side. This can break the bonds on the carboxyl side of an amino acid. At any rate, what I'm trying to get you to understand is that these guys are going to break small peptides down actually into individual amino acids, or maybe into little couplets of amino acids. So we started with big proteins, and we're breaking them down smaller and smaller and smaller with every step. And that's the main thing. You ought to be able to recognize anything with an ion here, pepsin, trypsin, as being a protease, breaking down proteins, an enzyme that breaks down proteins. And you know, again, if you can recognize that peptidase, that's telling you it's breaking peptide bonds. Okay, so the products ultimately here are amino acids. Now, nucleic acids, DNA, RNA, from whatever you've eaten, also get broken down. So nucleic acids get broken down in your digestive tract. What does the digesting? Or again, nucleases, again, that are also made in the pancreas, enzymes that are made in the pancreas, and the products that result are nucleotides. And then those can even get further broken down. We'll talk about that in a second. Alright, so we've done all of this chemical breakdown in the duodenum, okay, in the first part of the small intestine. Now we're going to head to the rest of the small intestine, where we're hopefully going to absorb. And what we will see is that the intestine, here is this long tube that is the intestine. It is not just smooth like the inside of the paper towel tube that we talked about earlier. It actually has lots of hills and valleys that are being shown here. This is essentially the same diagram. We're seeing the hills and valleys here. These hills and valleys are called villi, and if you'll note that on top of the, like here's a villi right there, but on top of the villi there's even smaller hills and valleys. These are called microvilli. And again, why do we fold up something? Because we're trying to fit it into a very small space. So microvilli and villi are ways to get a lot of surface area packed into a very small space. Why do we want to do that here in particular? Because we want to absorb as many nutrients as possible, and the more surface that the nutrients can spread out along, then the more of them we will absorb into our bodies. So there are actually some enzymes also embedded on the surfaces of these villi, microvilli, that are going to allows some of the very final breaking down to occur right before we absorb these nutrients into the bloodstream. This is the space where the nutrients are inside the small intestine, and we're going to absorb them across the wall of the intestine, and there's blood vessels waiting to pick up the nutrients and take them all around the body. Okay? So, we will do the last little bit of chemical digestion that needs to occur in enzymes on the surface of the microvilli, and then those tiny little monomers that your body is going to use for its own purposes will be taken by the bloodstream and off to do whatever they need to do. Okay? So, there are embedded in these microvilli here, okay, disaccharidases. Guess what they do? They break down disaccharides into monosaccharides, and those sugars go in the bloodstream. There are dipeptidases embedded in that membrane. They break down any dipeptides into amino acids. There are nucleotidases and the embedded, or actually I think they might be called nucleosidases. It's a chemical point that I don't think it's important that you understand, but they're going to break nucleotides into their three components: the nitrogenous base, the phosphate, and the sugar. So we're just breaking down into the smallest bits possible and putting them into the bloodstream, absorbing them and sending them into the bloodstream. We're going right across this membrane and into the bloodstream. This is a great chart kind of recapping for you where this is carbohydrate digestion, protein, nucleic acid, and fat, and it's telling you where everything happens. Essentially, the only thing that gets digested in your mouth is carbs. Essentially, the only thing that gets digested in your stomach is protein. But look, everything gets broken down in the duodenum, in the lumen space of the small intestine up there in the duodenum. And then we get a lot of further breakdown right before they cross the border into the bloodstream. This is a great chart to kind of summarize what's happening. This is also a good chart to summarize what's happening. Sorry, my kid just came in the room and distracted me. Anyway, this is a good chart to summarize what's happening as far as the different digestive organs. Groovin'. Feathers. You didn't kill a bird, did you?
No, there were feathers on the driveway.
Oh, well, they're pretty. I think they're woodpecker feathers. All right. At any rate, you might want to look this over and kind of just get this bottom line. And if there's something here that you don't understand, I think I've mentioned everything, then you can bring it up in class and we'll go over it. Did want to get this, make sure you remember, herbivore digestive tracts are way more complex than carnivore digestive tracts because that cellulose is so much harder to digest. We don't even have the enzyme needed to digest them, so cows and other herbivores have to harbor bacteria in their gut and keep these living cultures of bacteria going that will help them digest this very difficult to digest material. So make sure you understand that. Cow has essentially four stomachs, four chambers. You can see the food being taken in. It's going to go through here, and let me see, and then go out to the mouth again where it's going to be chewed some more and then sent back down in, and we're going to finally pass this stuff out and into the intestines so it can be absorbed. But there's a lot of digesting, breaking down that has to go on. This I wanted you to see so you could see the comparative length of the herbivore versus the carnivore and this complex thing called a cecum. It's a hugely complex organ that is partly there to digest that cellulose, that plant material. And I wanted to show you what's left of your cecum. This is your cecum right here, and your little bitty appendix is what's left of that very convoluted structure that was once there to digest all of that cellulose plant material. Your appendix is a vestigial organ. It once had a use. It has become highly reduced in size because we now, since we cook and things like that, we break down food differently outside of our bodies than and get more nutrition from meats and other things. We're not strict vegetarians, and so we have slowly but surely begun to do away with that part of our digestive tract. Okay, that's it. Um, and we're done. And thanks. And I'll see you too soon, I'm sure. Take care.